Agridos planipennis and Agridos anxius, first of all, they are two beetles belonging to the family Buprestidae, also known as jewel beetles because of their metallic coloration. And both of them can be very damaging to trees, their host trees, because of the larval activity. In fact, the larvae eat subcortical living tissues, so they can kill trees, especially when the tree is already stressed by other biotic or abiotic stressors. And Agridos planipennis is native to Asia, and it has been introduced into North America, where it killed millions of ash trees, its main host species. And it was identified as one of the most costly species ever introduced into North America. Both these species are listed in the European legislation, so surveillance and monitoring is mandatory for European countries. And usually inspectors exploit the different approaches, but one of the most common ones is the use of traps, so colored traps that can be set up at ports of entry and their surrounding areas. And these traps can be very efficient, especially for Agridos planipennis. The main issue with these traps is that they are not selective, so they are not specific to Agridos planipennis or Agridos anxius. So when we use these traps, a lot of native Agridos species are commonly collected. And so the sorting and identification to species level of all the specimens that are trapped can be very time consuming and requires a strong background in taxonomy. For this reason, any kind of technology that can help to identify all the specimens that are commonly trapped is really welcome. So this is the reason why we decided to apply the Entomoscope and associated algorithm to speed up this identification process.
The Entomoscope is a photomicroscope for capturing small objects in great detail. In FORSAID, it is used to train artificial intelligence models with standardized photos of insects for the purposes of automatic species classification.
So to start, we need to first select the correct lens for our specimen here. So for example, for this specimen, this lens with 0-point magnification is great. Then we need to turn on the device, the lighting system, and then we are ready to go with using Anima software. To begin, we need to first select the correct device. We have 3 different versions, but this one is this version. Then we need to do reference access to be able to start the autofocusing. We need to select first which fuse method we want to use, which we have a self-made method or a better one, which is helicon focus. We will move forward with that one. Then we need to select the lens, which is the correct one, and then we have autofocusing feature. Right now the camera is going down to find the correct lens and distance of the lens. Now we can also adjust the lens to be sure that we have all the details and information, which is, I think, is good right now. Then we have 2 options. We have we can take single image, or we can take stack images or multiple images and then stack all of them together. To be able to use this Anima software for real-time identification, we need to process these images. So we have different options here. So the first one is something extra, which is enable drying escape bar, something else. But we need to enable cropping methods. We developed 2 different methods, one which is fast method, the other one it takes more time but it's more accurate. And we can automatically detect the insects and crop that insect from the image. Sometimes, as you can see, we have really bad background. So in this case, we can also use this uniform background. We can define any color that we want, and we can automatically, using deep learning models, to apply it on the background. Then we can just start the whole process by using, for example, the stack images. We need to define the name here. So the camera moves, take 5 images and trying to stack all of the images together to have a really sharp image. And we are going to see the stack images here, right? This one. If we like it, we can continue with that. Otherwise, if you think it's not really good, you can just delete the last image and take another image. Meanwhile, you can see we are doing something here, which is doing all this processing in the background. At the same time, we can also press this measure specimen button. And here we load a machine learning model to automatically measure the specimen. So we can use this method. This method is going to draw a bounding box around the specimen, but also consider the orientation of this specimen. And then by knowing all of this camera information and lens information, we can convert the pixel to millimeter to calculate the length and width of the specimen. So the model detected the specimen. We have also the correct positioning here, but we have human in the loop supervision. So you can just adjust the bounding box here if you like. Maybe you want to just measure one specific part of the specimen or anything else. So you can use it. If you like it, you can just press confirm and we can automatically measure the length and width in millimeter. If you like it, you can just press save annotated image and move forward. Now the processing also done. You can see all of the images here. We have original, cropped, uniform and cropped uniform images. And then here we have a flexible inference engine. You can load any pre-trained model. As an example, we already loaded this model here, which is an initial model for Agrilus. Then you can select your model based on in which kind of dataset you train your model here, and then by just pressing classify, it will load the model, process the image, and you can see here we have this prediction with this probability, which is correct actually. And we can save all of this information locally, but also if you would like, you can use Zenodo upload here, which is you can just upload all of them in your desired repository on Zenodo. So you can see here, this is more automated workflow for insect digitization. Also, to be able to use it for high throughput analysis, we developed some plugins here. We have all the functionalities here that I explained. For example, for classification, you just need to load this plugin and you can load your pre-trained model, you load your dataset, which could be thousands of the images, and use this function separate from the animals or software and just process all of them at the same time.
The Entomoscope could be a very important scalable tool that everyone can use, since it's very easy to use and it's quick and it's reliable, since we reached high accuracy with it. So it's something that, if used properly, can speed up and save a lot of money and time to everyone.
We want to expand AI capabilities and to include more species to be identifiable. This can be done by integrating molecular biology, combining DNA barcoding with imaging, and expanding the range of application within Europe or even abroad, for new specimens. And in the next step, we also want to enable portable version that can be used for field work and to do identification just on site, near a trap or so,